You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Fatima AlZahra. She's recently formed a startup that uses some of the research that she was involved in bacterial communication. It's a prediagnose, is the name of it. So we're going to be talking about bacterial communication and her startup and everything else. So Fatima, thanks for coming. How are you doing?
How are you doing? Thanks, Richard.
Yeah. So tell me, what was your work like, when you were in the research area, and why did you focus on bacteria? Let's start with that.
I heard once I started at the university that bacteria talk together, and I thought it was extremely fascinating that they had this language that they could use to coordinate their activities. So I was still a very young bachelor student, and I thought maybe I could use it for something sometime when I have learned enough to make my own research subjects. So it stuck with me. It was so interesting that I kept thinking about it throughout the years, and then I thought, why not try to detect these molecules, these communication molecules that bacteria use to talk to each other, to make diagnostic tools to diagnose infections early. And that's how it started.
Very, very interesting. So in general, what do you believe that bacteria, I guess a simple word is to call them smart. How sophisticated do they appear to be now that you've studied how they communicate?
It's really interesting that they seem smart. I don't think they are conscious or anything, but it's the way they coordinate their activities that make them smart and make their world a bit parallel to the human world. So the idea that some of the activities they have, we can draw parallels to the human world. That's the smart thing about it.
So what's an example of how bacteria would communicate? What would happen? Like a bunch of bacteria would be exposed to some kind of environmental stress, and they would communicate about the stress. What's an example?
So sometimes it could be external factors that make them coordinate their activities. But for example, they all need to produce some common goods for the society, so they all survive. That's the main thing about the collective behavior. So they don't exhibit individual behavior, because that will not really benefit them. But when they do it all together, that will benefit the whole community. That will make them survive as a community. And if we draw parallels to the human world, then it's actually just like how we build our society. If we live alone, we will not really be able to do anything. But if we help each other, some of us are doctors, some of us are teachers. We build a community that can make the whole society thrive.
Well, for bacteria, what about experiments with a single bacteria in a culture medium that's exposed to an environmental stress? I would think that the bacteria do want to preserve themselves individually and not just die and give up. I thought that they evolve and respond to threats and rearrange their genes and do all kinds of stuff to at least keep homeostasis, if not try to preserve themselves from dying.
Mm-hmm. Yeah, is
that what you've observed?
Yeah, they can have a lot of strategies. It really depends on which bacteria we're talking about. But yeah, sometimes they form biofilms, and then they can hide inside this biofilm and avoid being exposed to this external stress from the surroundings. And there are many strategies they have.
So, in order to have a strategy, I mean, wouldn't a bacteria have to be self-aware? And then even to communicate, wouldn't it be, itself is communicating with something that is not self, with another bacteria? So wouldn't that signal some kind of, awareness or intelligence?
Yeah,
able to do that?
I wouldn't say that at all, because from a biological perspective, it's more about chemistry. So they release some molecules that other bacteria have a receptor to receive, and like convert to another signal that they know how to respond to. So it's not self-awareness in that sense that we understand from humans, where we really reflect about our decisions. It's really a chemical process, biochemical process that is going on with these creatures. But I don't know in 100 years maybe someone will come up and say they are conscious. I don't know.
So what are some examples of the communication that goes on that you've observed? Any specific examples?
Something I worked a lot with is their expression of virulence factors, so factors that will harm the human body and maybe also harm other kind of bacteria or other families of bacteria. So they could the bacteria I was looking at could survive and dominate the culture they were in, and they do this by exactly releasing these communication molecules that will tell the other bacteria that they are enough to secrete a toxin that is toxic to the human body, but also toxic to other species.
So, I've heard of like quorum sensing. Bacteria will sometimes not engage in a behavior until there's quote unquote enough of them, and they are sending out signals.
Yes.
So can you talk about that?
Yes, that is exactly quorum sensing. So they release some molecules, and they all understand that they use them to understand the density, how many bacteria are they right now in this particular situation, and based on that they can all secrete a molecule at the same molecule at the same time. So this way they can synchronize their behavior.
Okay. So what kind of forms of communication are they? Like, again, one form of communication, I guess, is identifying or putting out chemicals that only that particular strain will be able to note came from another one of that strain, of the same strain.
They could have a global language. They could also have a language that is only understood by the specific species. So they use that to coordinate what kind of molecules they should secrete in response to that.
Okay. Do you see that bacteria are communicating with other strains, or is it mostly with their strain? How do they interact with?
They could do it across. There is something called the cross kingdom. So they release the molecules across their own species, so other species understand them, and then they have the secret language where it's only the species themselves that understand what is going on and how they should respond to it.
Oh, so all right. So they'll communicate in different ways.
Yes.
In one way with their own strain, and then in different ways with other strains.
Yes.
How many different strains can a particular bacteria talk to? Has anyone tried to look at that and see if they can communicate with dozens or hundreds of other kinds of strains, or is it limited to a few very closely related ones?
There's something called quorum sensing systems and something called the quorum sensing regulated molecules, and it really depends, and we haven't even discovered everything that is released out there. But what I have been working on specifically is to detect these molecules. So I have developed the sensors to detect these particular molecules and try to see if we can use them to understand something about the colonization or the infection.
So what size are the typical bacteria you've looked at, and then what size are the chemical messengers they send? Do they just send out naked chemical messengers, or do they enclosed in a vesicle, with a membrane, and they're absorbed into the receiving bacteria? Like, how does this communication look?
I've only looked at the extracellular molecules, so it's molecules. I don't look at sizes at all. I look at concentrations. So I try to understand what is the concentration that they secrete of a particular molecule.
Well, I mean, we can observe, some bacteria at least under, a microscope. So I just wonder if their communication can be seen under a microscope, or if it's so much smaller.
Yeah, I
the
think it's much
extracellular
smaller. I think it's much smaller, but some of the behavior they exhibit, it could also be seen with the bare eyes. So, if, for example, one of the molecules they secrete, it turns the whole culture green, and that is something you can just see, and it happens instantly.
Huh.
So it's really fun. Sometimes you're in the lab and then you see a culture just turn green after a specific period of time.
Okay, so what have you been able to figure out by the density of the molecules that are being put out bacteria? Is there a lot of crosstalk, or is it do things go quiet for a while and then there's talk? Are they constantly talking to each other or signaling each other?
Yeah, so according to theory, they should constantly be secreting these molecules, and then if the environment they are in, it's just a lot, and the molecules just flow away, then the other bacteria will not really sense it, and this quorum sensing regulated behavior will not appear. But if the solution is dense with these molecules, these quorum sensing molecules, then it will allow the quorum sensing regulated behavior to be activated.
Have you tried to take, let's say you have like a certain strain, and you take some of the fluid that they're swimming in into a little pipette, and then pipette that into the fluid that the same strain is living in another dish. So you artificially increase the concentration, or maybe change the concentration. Have you tried to do that and see what their behavior?
Yes,
how their
and
behavior changes?
yes, I've done that, and actually many others have also tried to do that, and it really depends on what time you took this solution to the further experiments. So sometimes they are just oversaturated with molecules, and that means that they stop producing something. The new cells stop producing something themselves because they think, or they don't think, but they sense that there is enough, and sometimes it's just not saturated enough, and then they still start producing it. So it really depends on the setup and the parameters you're starting the experiment on. But
sure. But what kind of behaviors have you been able to modulate by doing that?
I've seen that we can actually saturate the culture by taking a culture a supernatant that is full of these quorum sensing molecules, and then saturate it to a sense that the bacteria stop secreting some new molecules themselves. And that could be beneficial. For example, if you want to confuse the bacteria, if you want to somehow manipulate them to think or to sense that they don't need to produce any more virulence factors. But that's a whole other subject, and actually a lot of people, or not a lot, but many competent people are just focusing on that, how to use quorum sensing as an alternative to antibiotics. So by confusing the bacteria, by stopping their communication, they can stop the whole infection to proceed.
Oh, so I guess one way would be to convince the bacteria way early on that there's enough of them to mount an attack, so that they're much more likely to be defeated. And I guess another way would be to, if there are a ton of them, to block their communication somehow so that they stop attacking and they sit there and then get picked off by the body's immune system.
Exactly. So if they get blinded, or if they can't see that there are other molecules or other microorganisms nearby that can help them in this community they are in, then they just stop, or they get confused. They don't know what to do because they don't have this network that they are in. So that's another way, and it's actually a lot of research is going on right now where they try to use that and to as an alternative to antibiotics, because we're running out of good antibiotics in the future.
Do you see that bacteria communicate cross-strain, or do they only really work with their own strain?
It really depends on the setup, and it depends on the strains themselves. Sometimes the strains, they differ from lab to another, even if it's the same species, and the same strain type. So it really depends. There's a lot of research showing that sometimes they fight together, sometimes they cooperate. It's a lot, and it's actually really fun to see how they behave, the different strains.
Right, but sometimes they do cooperate with other bacteria that are not of their same strain, and sometimes they don't. Sometimes they fight them.
Yeah, definitely. Sometimes they cooperate, and that works just fine. And sometimes they need to dominate, and then they outcompete the other species.
So maybe another idea would be if someone has a bacterial infection, if you put bacteria that they would normally fight with, into the recipient, so that their attention is diverted from attacking the host to fighting with the other bacteria, maybe they would knock each other out and then not bother you,
Yes.
and not bother the host.
Yes, exactly. So tricking the bacteria into some sort of behavior that is beneficial for human beings is also very popular right now to look into.
Are there hibernation states or low or high energy use states that bacteria can exist in, and maybe that can be triggered? When someone's infected by a bacteria, they can be, again, like you said, tricked into going into a hibernation type state or a non-active state.
That's really difficult to answer, because it depends on what bacteria we're talking about. But some bacteria.
Oh, sure. It all depends. I know, but again, have we seen this in any bacteria that you've looked at or you've seen in the literature?
I haven't worked with that at all, actually.
Okay, all right. So, what's the focus of Prediagnose, your startup? What are you looking at, and what's the technology about?
The technology is about creating sensors that can measure these communication molecules and the quorum sensing regulated molecules to understand how bacteria behave during infections and during colonizations, and see if we can use that to, first of all, create early diagnosis of infections, but also to foresee how the infection is going to develop. So this is the core of what we're trying to do here.
Oh, so what's the normal process, as you understand it, of bacterial infection? What kind of things have you observed happening? What are the steps, for instance, and how do you imagine intervening?
So right now, if you want to get a diagnosis on whether you have an infection or not, then you need to go to have your sample cultured at a hospital or at a clinic, and they send it for laboratory analysis, and that takes time before you get an answer. And sometimes you also need the bacteria to get up concentrated in your body before you can even see it in a culture. So what I try to do is to make instant detection. So whenever you suspect an infection in your lungs, for example, or in your blood, then you can give a sample, and then 30 or 60 seconds later, you get an answer on the spot.
Well, how come you would be able to do it and traditional culturing methods, need a ton of bacteria to do it? What makes this more sensitive and faster?
I work with a completely other technology. I don't need to culture the cells first. I work on detecting these molecules that are secreted by bacteria, and the sensor itself is so sensitive that it can catch those small bacteria that are secreted in a sample. And that's why I can detect it much faster, because I don't detect the bacteria directly. I detect the molecules the bacteria secrete.
Well, if a bacteria infects a person, the bacteria is so tiny and the person is so big, even if they're secreting molecules, how would there be enough concentration as it disseminates and filters throughout the whole body to pick that up? Do you have to kind of know that, okay, this person may have a lung infection, so we're going to
Exactly.
sample
So
right near the lung?
Exactly. It wouldn't work on any patient. It would work on patients that are immune compromised. So it's patients that we expect are vulnerable to infections, and they are already followed by doctors because it's important to catch any infection up front and treat it early. Otherwise, it can really culminate in, for example, a chronic infection. So these patients are already monitored, and if we can follow them early, if we, for example, suspect that they would have a lung infection, then we measure in the sputum of these patients. If there are other group of patients that are in special risk of having a blood infection, then it's the blood we monitor. So we try to detect in the focus of infection that we expect.
So if you're able to detect it much faster or earlier, what are some of the ways that you could intervene that we can't do right now?
That would be to detect these molecules faster than it's possible today, because now you need the bacteria to grow on a plate, and I don't really rely on that. I rely on the molecules they secrete.
So you're hoping to catch them earlier in a less powerful or weakened state or less numerous state, so that even the current medicines out there might be more effective against them.
Yes, exactly. So I hope to catch them during this window of opportunity, as the hypothesis is that that in a very very early stage it's possible to treat them with antibiotics, with even less antibiotics, and in a stage where they could be easily killed with medicine, instead of waiting until they are really resistant and tolerant and can't really be eradicated.
So what kind of conditions are you targeting? What kind of bacterial infections?
I've been looking on some specific bacteria, and one of the bacteria I've been working a lot with is Pseudomonas aeruginosa. We're expanding to other bacteria of interest for healthcare.
Oh, what does the Pseudomonas do? What's the effect? What does it cause?
It's an opportunistic bacteria, so it is dangerous for people that have an immune compromised system. It's a bacteria that is actually everywhere. It's in the environment, so it wouldn't really affect healthy individuals. But for immune compromised patients, it's really risky to have this bacteria.
Okay. And what does it cause? What kind of disease does it cause? It just kills the
really
person, or
depends.
what does it do?
It depends on where the infection is. If it's in the lungs, it could cause really, really nasty lung infections. If it's if it's in the blood, it could cause sepsis. If it's so it really depends on where the patient gets the infection. But sometimes sometimes it's related to the contact lens infections for healthy peoples, but but at that point it wouldn't be that dangerous unless it isn't treated in time, and then it can be really dangerous to the site.
And how do you think the like our immune system interacts with invading bacteria? I mean, I've known of like T cells engulfing bacteria, but do you think that our immune system are sensing the chemicals that are given off by bacteria just as if they were bacteria, and reading that and interpreting it, and then knowing that they're out there and they need to be attacked?
Yeah, usually the body can recognize pathogenic bacteria and even the opportunistic bacteria. So that wouldn't be a problem. It's only when the immune system is defect that it can, for example, for the opportunistic bacteria, it can really cause damage to the human body. And that's why it's important to detect them early.
Well, I mean, what's the method by which the immune system detects these bacteria? Do you think it's the same way as a bacteria detects another bacteria, by looking at these chemicals that are given off by a bacteria and evaluating them?
No, the immune system works in another way than how the bacteria recognize each other. It's a whole science itself, but that's actually not my expertise.
Well, the reason why I ask is, what if you were able to create a vaccine just based on the not on weakened or dead bacteria themselves, but just on the chemicals that they give off? What if that was to be used as a vaccine? Would that work, do you think?
I think that there is some research going on where they try to trick the immune system to understand these molecules that the bacteria secrete. But I don't know how far they are with this. It's really preliminary research.
Okay. I mean, yeah, what you're studying could shed light on a lot of ancillary topics. That's why I'm asking.
Sorry?
Well, yeah, what you're studying could shed light on a lot of ancillary topics, on a lot of other,
for sure.
Other possibilities.
Definitely. It could open up for a whole new way of studying bacteria, even because now if we have this high throughput method that I'm working on through prediagnose, it would allow another kind of research, because right now you need an HPLC to conduct thorough research where you really pre-treat all of the samples and try to go through the pre-treated samples one by one through a heavy machine, but doing it very fast within few seconds, that will allow us to gain a lot of information.
Okay. Why work with immune compromised people? I mean, I would think that there are bacterial infections that overcome even healthy people's immune systems.
Yes,
do you think there's just less factors involved?
No, it's just to begin somewhere, and it's really important to do because immune compromised patients are really in risk when they get these infections. So we started with the immune compromised patients, but the vision is that we can expand it to a lot of other just actually healthy individuals, and maybe not only look at pathogens, but also look at healthy bacteria and see if we can figure out a way to describe a healthy a microbiome in the human body by looking at some signaling molecules.
Okay. What about phages or the body's microbiome? I mean, I guess that complicates it. I don't know how that changes the interaction of an invading bacteria.
It does.
Are you able to look at that, or is that just?
No, not yet, not at all. But we it's still in the research and development phase, and we hope that at some point we can do that.
Okay. All right. Well, very good. So, what's the best way for people to learn more about your work and maybe to get in touch with questions or ideas for collaboration?
I am active on LinkedIn and on you can look at the website prediagnose.dk, but you can reach out through LinkedIn and I would be happy to answer questions.
Okay. Well, that's great. Well, Fatima, thank you so much for coming on the podcast. I appreciate it.
Thank you, Richard.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.